Did I share it? No? Oh.
Did you click on the share screen? Yes, you are sharing your screen.
Am I? Just because I just a second.
No, yes, you are sharing your screen now. We can see your presentation now.
And just a second. Now it's okay.
Okay, can you see the number 76 down on the bottom of your screen? Yes. Okay. No, no, don't touch that. Near that, just no. The one. No, no, the one. Okay. Yes. That. Just touch that.
Oh
Yes.
yes, that one.
Yes,
It's
that one. Yes. Great.
so I just to check how it goes. Now it's okay. So thank you very much for the opportunity to share you my research work with the team of Professor Dulashka from the Bulgarian Academy of Science. So we know that Saccharomyces cerevisiae has been known by mankind for millennia, not only for its application in baking and brewing. Today, I'm going to talk about the Saccharomyces cerevisiae as a model organism, more specifically about its behavior under oxidative stress. I will tell you later about application of the Saccharomyces in the finding and functional units of hemocyanins of Haliotis tuberculata. So why we have chosen Saccharomyces cerevisiae? Firstly, we know that the Saccharomyces cerevisiae is not pathogenic. Recently, in the past decades, the full genome of the Saccharomyces was fully sequenced. And it is also known that the yeast genes have orthologs in the genes in the higher eukaryotes. What are actually the advantages of the Saccharomyces cerevisiae when it is in quiescent state, in a G0 state as a model organism? We know, and it is known for a lot of time, that most eukaryotes spend their life cycle in the state of quiescence. And the majority of the cells, if we compare it with human tissue, the majority of the cells in the Saccharomyces cerevisiae in quiescent state do not undergo division. Prokaryote and eukaryote microorganisms in quiescent state survive for long periods of time, even decades and centuries. What we do here is that we know that biochemical processes normally they lead to the formation of reactive oxygen species. And the cells normally are capable of balancing the production of these ROSes, but not all of them are valiant for them. When there is more of these ROS and the antioxidants, they cannot they're not capable of scavenging the ROSes. The ROS they cause oxidative stress. It means that we can cause damage of the cell or even death. How actually, why and how the cells of Saccharomyces cerevisiae entered in a quiescent state? Actually, they're grown in some nutrients, predominantly glucose, and we know that glucose they use the glucose in order to produce ethanol fermentatively. After the production of ethanol is finished, they transform it through respiration, and then we are over. We don't have the ethanol, and after that, we got the the Saccharomyces cerevisiae enters in a quiescent state. What is the characteristics of the cell in general in a quiescent state? They do not reproduce. The protein synthesis is really low. They become more thermotolerant because of their thicker cell walls. They become more stable against toxic compounds. They are characterized with increased response to oxidative stress, and they get more bigger reaction in the signals to the signals from the environment. What we have used in order to prove our to make our experiments, we have used yeast strain Saccharomyces cerevisiae, which is not it's diploid strain obtained from the Euroscarf collection. We have actually not our team, because we don't our team from the biological Sofia University Department of Biology. They have cultured our cells, and they have made predominantly tests for the LD50 doses of the following agents. So we have treated the cells with hydrogen peroxide, menadione, ibuprofen, and zeotine. They have determined these concentrations, these LD50 doses, and after that they have extracted the material. Actually, these are the steps we have gone through in order to make our analysis. As I said, our team from the Sofia University Biological Department, they have cultured the cells, and we have received some cell homogenate from the Saccharomyces cerevisiae. We have a control, which is not treated, and treated one. After that, we have after centrifugation, we have received the supernatant. We have used it to make sodium dodecyl polyacrylamide geoelectrophoresis. After this sodium dodecyl polyacrylamide geoelectrophoresis, we have chosen the bands which are different. So where there is a change in the expressions in comparison to the non-treated cells. After this, we have made denaturation of the proteins of the bands. After that, we have treated the extract with trypsin. And after trypsin digestion, we have received the peptides. These peptides we have measured through mass spectrometrical analysis on autoflex Maltitov systems. The received data, which are masses, we have identified through the database in matrix science through mascot analysis. So here is our results from the SDS page electrophoresis. So here we compare actually two phases, the proliferating state and the quiescent state, treated with different oxidants, as we said before, hydrogen peroxide and so menadione and so on. And extracted peptide fraction, this here mass spectra is the spectra of extracted peptide fraction after trypsinolysis of lane 2, some here, of band 2, it is here, from the electrophoresis. So what we have done, this data, these peptides were inputted in this is the database mascot peptide mass fingerprint. We have put it in the query segment. After that, we have received some putative results, and the results that have more higher score, we have chosen them. Higher score, we mean when the protein sequence coverage is higher than 15%, let's say. The following table gives us information of all the identified bands of the electrophoresis, bands which are of our interest, where we have found changes. Some of them are cytochrome C peroxidase, DNA replication fork, NADP dependent diflavin oxidoreductase, and AP1 like transcription factor, 5, YAP5. So the next, we have used some Saccharomyces as enzyme who will degrade the big molecules from the hemocyanins of Haliotis tuberculata. And firstly, I will tell you about the hemocyanins. What is a hemocyanin? They're copper-containing glycoproteins, which are with a complex quaternary structure, responsible for transferring oxygen for the hemolymph of mollusks and arthropods. In the past decades, the hemocyanins are object of big interest because of their high molecular mass, differences in the oligosaccharide structures, and their immunological properties. We throw light upon Haliotis tuberculata because it is really found that it is substitute, a really good substitute of the hemocyanin of Megatura crenulata or the keyhole limpet. It is known about this hemocyanin from Megatura crenulata that is applicable in the as medical immunological product. It's called Imucotel, and it's used in the bladder cancer immunotherapy. So these macromolecules, the hemocyanins, their ring form decamers with molecular weight over 4 megadaltons. Hemocyanins from Haliotis tuberculata. Haliotis tuberculata actually is the green armor. They're represented with two isoforms. Subunit 1, HT1, it's presented here, and subunit HT2. So the big difference is that the second isoform can form D-decamers. What we have is every subunit, which is here, actually, just a moment. We have a subunit which is represented here. It is represented from 8 globular structures, which are called functional units: A, B, C, D, E, F, G, and H. It is known for them. Our team had made some models of the functional units of the Haliotis tuberculata. Okay, so what we have here is the core, where we have the potential and linked sugar trees. for all of the 7 functional units except functional unit HT1H, which has 2 potential end-linked sugar trees and 1 cupridoxine tail, which is here with the blue dotted line. It is circle. Our team has also analyzed the glycans for which are found in the functional units. Up to now, 15 carbohydrate structures, which are predominantly composed with high monols composition. What is our hypothesis actually is that these glycans participate in the formation of the connection of the different functional units in between with the carbohydrate structure. They are included in the composition of the subunit. What we have used actually, this is the functional unit A, B, C, D, E, F, G, and H, and in between we have this carbohydrate structure which connect them. How actually scientists in this area, how they prove? They use enzymatic hydrolysis with trypsin, with PNGase F. But our method, we have used a new method, incubation with Saccharomyces cerevisiae. That is why I told you that Saccharomyces cerevisiae we have used as for enzymatic hydrolysis. And it is known that they produce specific glycosidases that will destroy these glycosidic bonds, and after that we will receive these functional units. In order to prove our results, we have used another enzymatic hydrolysis with a really simple enzyme, zymoliase, so it is really with a high concentration. Then after that, after this enzymatic hydrolysis, we have isolated the functional units. And for that, we have, in order to determine their molecular masses, we have used Mauditov MS analysis. And after that, the results we have compared with the work of other 2 authors that have worked on this topic. So here we have the results of the 2 isoforms, the HT-H1 of the 2 subunits of the hemocyanin from Haliotis tuberculata. And here we have presented the electrophoresis with incubation with Saccharomyces cerevisiae, all the subunits. So we have a non-treated subunit 1, treated for different for 24 hours and for 48 hours. And again, from lane 7, which is here to the end, so we have the non-treated subunit 2 and the treated subunit 2 after 24 hours and 48 hours. And in the end, we have the molecular weight protein marker here. In order to analyze the electrophoresis, so we have used a software program, ImageQuant TL, which has helped a lot. So what we observe here, so I present the results with for the functional units. If we observe lane 9 here, so we observe that the masses of bands 11, 12, they, after that I will show you the comparison with the results from other authors. 52 and 52 and 46 thousand daltons is really acceptable in comparison to other results from the other authors. So in order to prove our results, as I said, we have used another enzyme, zimuliase. And here actually we will receive better results for subunit 1 and subunit 2, because in the first with the incubation with Saccharomyces cerevisiae, we did not receive so good results for the subunit 1 of the hemocyanin from Haliotis tuberculata. And actually, here we have received data for functional units with masses 46 and 51,000 daltons, and for subunit 2 we have received 47 and 52,000 daltons as molecular weight. Actually, we will see after that that in electrophoresis, one bond can have more than one bond can contain more than one protein, and in the databases, it is known that most of the functional units, they have mass around 46,000 daltons or 46 kilodaltons. That is why we'll see here like 2 molecular weights in the results. Actually, Leap and Keller et al. Had used other methods to determine the molecular weight, and they receive data which is really close to our results. What we need to do now is after preliminary determining with the software MHI-QTL the molecular masses, now we have to make the TRIPS analysis and to make the analysis with the mass spectrometric analysis in order to determine the masses of the bands of both subunits and both enzymatic treatments. So this is from me. I would like to thank first to my work team, especially to Professor Pavlina Dulashka, to Associate Professor Ludmila Velkova, and our research group from the Sofia University Biological Department, Professor Anna Klimjieva, Professor Ventislava Petrova. And of course, to the research work was supported by the National Science Fund under grant agreement DN11 from the 15/12/2017. Thank you very much for your attention.